somehow it's figured out a way to make its way into my cells, punch a hole in them, go and live in there, start dividing, and completely avoid being cleared by the immune system forever. And I was like, so this thing, I feel like in some way, knows me better than I know me.
Not as dumb as it looks.
No. Hello, everyone, and welcome back to People, Parasites, and Plagues, a podcast aimed at delivering information about the fascinating pathogens among us from the scientists who study them. I am Kim Klonowski.
And I am David Peterson, your hosts for today's episode. If you want to starve a parasite, the first step is to understand how it eats.
Today, our guest is Dr. Drew Etheridge, Associate Professor of Cellular Biology, a member of the Center for Tropical and Emerging Global Diseases. Drew, it's great to have you with us.
Great to be here.
So I guess the first question we like to start with is what we call our pathway to science question. Take us through how you ended up in a career in science, and you can start in kindergarten, whatever's relevant, first grade, even high school for that matter.
Well, I believe it started when I was very young. My father was a scientist, so he ran what we would call, a service lab in the poultry science department. He was trained as a chemist and a nutritionist, but he was a natural scientist. He would always do experiments around the house, and he would breed fish. He would always graft plants together, and he was a big hunter, and so he would also breed bird dogs. And so he was always doing weird experiments around the house and documenting them, extensively. And so science was always kind of something that we would talk about, and he would always humor my interests as a kid. And there was never too many questions really for him about how something worked or why something was the way it was. And that was always fostered at our house. So I think that probably played a big role in it. He was more of a natural scientist. I feel like I like to deconstruct things and take things apart. And that was always fostered. So I was originally kind of into chemistry when I first got into college. And I decided to take a biochemistry class. And then I said, well, this is way more interesting than chemistry, from my opinion. And that kind of started my interest in looking into biology labs and doing experiments in something independent or different from a chemistry lab where we're synthesizing stuff and doing chemical reactions. So my first lab I worked in was here at UGA in Harry Daly's lab.
Oh, yeah.
Who studied heme biosynthesis. And one of his graduate students, Amy Medlock, who's now a professor here,
right,
was my mentor at the time. And so she taught me a lot of really cool stuff about what was kind of at the time very cutting edge in the mid-90s, which was molecular biology, being able to clone genes. And I remember being kind of just taken by that as I'd never seen anything that cool before. And I remember going home to all my roommates in college and being like, you cannot believe all the things we can do. You can take a piece of DNA out of one organism and put it in another and then express that in a bacteria and then purify the protein and do experiments with it. That was amazing to me. So, I think I've always been interested in how things work. And I think biology to me, and this is kind of the way I talk about it to my classes, is biology is really just a massive reverse engineering project where we're taking these extremely complex machines apart and trying to figure out what the individual components of these machines do biochemically. And that was, always, something I found fascinating. But I had other interests, but I wasn't sure that I wanted to do a full career in science, even though I got an undergrad degree in molecular biology and biochemistry. Wasn't really until I kind of came back to UGA after spending some time in Europe and started working as a technician in Rick Tarleton's lab that I really realized that this was something I wanted to do. Because I had great experiences in various labs. I worked in Harry Daly's lab. I worked in a guy named Lars Youngdahl's lab as an undergrad. But it was Rick's lab it was a lot of fun. Like I really enjoyed it, and I liked the style. He was very tolerant of a lot of my antics and questions, and allowed me to kind of express myself.
So was that your first exposure to parasites, to studying parasites in Rick's lab?
Right. So I worked, in various kind of biochemistry labs. When I first saw, so oddly enough, T. cruzi was my first kind of parasite I'd ever seen. It's a very vivid memory still when I first saw kind of an infected cell before. And I don't know, it was very strange feelings that it brought up. I remember being like, oh, this is monstrous. Like, this is kind of disgusting. It's kind of these wormy things inside of a human cell. And I had never seen anything like that before. And I was like, this is crazy. And then realizing, well, once you get it, there's nothing you can do about it. It's like, can't fight it, and you have it for life. And I was like, oh my God, like this is a monster. And
is that when you decided to stop mouth pipetting in the lab?
That was when I started wearing gloves and a lab coat.
Right.
And no, it had a profound effect on me. But I also realized, as a tech in his lab, how little I knew too about, because there were things I was like wanting to do. I was like wanting to get better at molecular techniques, better at biochemistry. And I realized the only way for me to do that was to go to graduate school. And that really is what set that off, that experience in that lab. And I wasn't necessarily sure that I wanted to go into parasitology. Part of me wanted to go into immunology. I found that to be fascinating. When I ended up going to grad school, I rotated in, 3 immunology labs. And none of them seemed to be a good fit or had the skills that I wanted to learn specifically. So I ended up going into a parasitology lab again. And instead of T. cruzi, it was T. brucei, its cousin. And so, and that's kind of how I started down the path in parasitology. It was, what I say is it's a lot of serendipity, a lot of chance encounters. I would be lying if I said that parasitology is what I always knew I had to do at all times. Like, there were a lot of scientific things different disciplines I'm interested in and topics. So it wasn't necessarily, it didn't have to be parasitology.
And that's interesting, because it seems like when you looked in the microscope that time, the response was so visceral that, I felt like you were going to tell us like that was what hooked you in parasitology.
Well, I mean, it did, but I think it also strikes at really fundamental ignorance about biology in the sense that you have this single-celled creature that on its surface looks really dumb, right? Right. You're like, oh, well, it's just a single cell. Like how, sometimes it's round and sometimes it has a flagellum and swims like a little worm. But you don't think much about the kind of the innate intelligence that's encoded in that creature. And I'm like, okay, so this thing somehow has figured out how to cycle between an insect vector, lives in its guts, somehow it's figured out a way to make its way into my cells, punch a hole in them, go and live in there, start dividing, and completely avoid being cleared by the immune system forever. And I was like, so this thing, I feel like in some way, knows me better than I know me.
Not as dumb as it looks.
No.
Not dumb at all, right? No. And so that was I think that was the thing that struck me, because then it started just making me think a lot about living things and biology in general in a way I hadn't thought about it before. Like, there's an incredible intelligence encoded in these things that kind of seem, stupid on the surface. And that was infinitely interesting for me. Immunology was a big part of that lab too. And so that was its own universe of cells that have different jobs and functions about attacking, invading pathogens. And I never really heard of such things before that time, that you have all these cells that are crawling around in your body looking for things to fight all the time. No one really talks about that, how much stuff is going on in the background to kind of keep one healthy.
Yeah, you're right. We need to talk about that more.
Oh, all right.
So why don't we kind of jump forward a little bit and talk about some of your current research projects? So what would you say is one of the main questions that you're asking in your lab with your research? I know you have your hands in a lot of areas, but...
The main thing we're doing is trying to understand endocytosis in Trypanosoma cruzi. So Trypanosoma cruzi is different from the other parasitic trypanosomatids because it has this ancestral structure we call a cytostome-cytopharynx complex that it uses to endocytose. And it's really just like a mouth and an esophagus that it engulfs things from the world around it and eats those things and digests those things. Whereas its cousins do all this through a structure they call the flagellar pocket. And they lost this. So Trypanosoma brucei and leishmania lost this structure. And so, we started studying this, again, like many things I've studied, by chance. Mm-hmm. And I had been interested in getting back into Trypanosoma cruzi research when I joined the faculty here at UGA, where I was originally studying toxoplasma and secreted effectors. And so I was curious to see if I could identify any secreted effectors in Trypanosoma cruzi because none had been identified, because I was curious about how it might be manipulating its host cell when it invades. And so we randomly picked a few different genes that might be secreted that looked like they might have signal peptides. And CRISPR had kind of recently become in vogue in T. cruzi. And I was like, well, maybe this is a good time to cut my teeth on seeing if I can do some transfections, see if I could tag some genes, which was at the time a very rare occurrence. And so I got Teresa, who was a postdoc in the DeCampo lab, to train me how to use CRISPR. And I just started tagging random genes that I thought might be secreted. And one of those genes I tagged ended up localizing to this very strange tubular structure inside the cytosol. And that led to kind of the discovery that was a first protein that people had ever identified that went to this cytostome-cytopharynx structure. And then I was like, well, here's a big gap in knowledge. And it wasn't anything I'd ever studied before. I didn't know anything about endocytosis before. But, we realized, no one knows anything about how this thing works mechanistically. And I think more importantly, no one knew how this worked in any protozoan. So ciliates, many free-living ciliates have this structure, free-living flagellates have this structure. But no one knows how it actually brings in, where the force drive comes from to bring in that material. Because it's a very strange structure. It's not kind of your classical endocytosis. It's a stable tubule that brings in nutrients to endocytose. And this is how ciliates capture bacteria to endocytose.
I'm just picturing the straw. I mean, can you visualize it?
I mean, you can see it.
Yeah.
Yeah. It starts as a opening at the surface of the cell, and it descends as a stable tubule. And then at the very end of it, you have vesicles that bud off and then go and fuse with lysosomes or for digestion eventually. So it's a stable straw in a sense, but it's also constantly moving. So membrane is kind of being pulled in and grabbing things with what we think are receptors there, pulling them down into the tubule. And at the end of the tubule, it's budding off.
So that is different from ciliates because they have their mouth, if you will. I think there's little cilia around it kind of directing things in. Am I correct there? Or
Right. So they help overcome, I think, diffusion by, in a sense, filter feeding in a way, by fanning bacteria towards their cytostome. Some research has suggested that there are various lectins receptors that the ciliates like Paramecia will put on their cytostome and that will bind to glycans on bacterial surface cell walls to help kind of facilitate capture and endocytosis. So yeah, they use, tons of ciliates do this. They'll use the cilia to kind of filter feed essentially. But the engulfment occurs through a cytostome.
Okay. But T. cruzi has, as you say, probable receptors around the tubule, and those get kind of, through a motion of I'm trying to explain this, and I'm
I
failing.
almost am thinking if it is like, I don't know. I'm sure these things have a name. I just don't know. Some of those trucks that have that kind of belt that kind of just goes out and comes back in.
It's like a conveyor belt. It's like
you know what I'm talking about in the trucks. They kind of, or those
like tank
things.
tracks.
Yeah, kind of.
I mean, we often refer to it like a conveyor belt in the sense that a membrane comes from what we think. This is how we believe it works. Membrane comes into the flagella pocket, gets pulled up and over to the surface, captures material, and then gets pulled down into the cytostome entrance. And then, so what we found is after we first identified the original protein and started getting into it, thinking about it, we had known that an actin polymerization inhibitor, cytoclase D, will block endocytosis. We didn't discover that. It had been seen a decade ago. And so we said, well, if that's true, then maybe myosin motors are involved. And so looking at the genome, Trypanosoma brucei and leishmania are operated like natural knockouts of the cytostome for us. So we started just randomly tagging all of these things that were originally classified as orphan myosins. And so what the parasite seems to be doing is coating the tubule in like an actin filament network. And then adjacent to that, there are these structures we call rootlet microtubules that it arrays a large swath of myosin motors that are grabbing the tubule and pulling it down like they're like the conveyor belts pulling the tubule down. And so we've spent a lot of time trying to understand how the tubule gets endocytosis pulled in. And so we found, a bunch of myosin motors that contribute to this process. And it seems that actin is being coated on the tubule, at least as our current model suggests, and that this is bound to the membrane of the tubule, and that the myosin motors are actually grabbing onto that and kind of pulling it towards the center of the cell.
So is it just bringing in individual molecules, or are we talking macromolecules that are being brought in? And do we know the identity of any of those?
What we've seen, so we've done some lipidomics of parasites, and we know that when we grow them in media with, say, FB, fetal bovine serum, that they're pulling in those components, those lipids from the serum. What's interesting is we've tried to see if they do have any specificity for the things that they endocytose. The cytostome is quite small, so we haven't really been able to show that they can pull in very large objects. But every protein that we've tried to feed them, they can endocytose that protein. So they seem to have a broad, non-specific type mechanism to bring in. So we've fed them BSA, we fed them IgG, we fed them a whole array of proteins, and it seems to be able to eat all of them. There's nothing specific about that. Whereas you would think, their cousin Trypanosoma brucei has very specific receptors for certain things. We thought maybe it would have something like that, but we haven't been able to detect anything like that.
So T. cruzi is an omnivore.
Right. It appears that way.
Right.
Yeah, there's no specificity that we can see.
That is interesting. How does it grab anything?
Right. That's a great question. We have found one thing that appears to be a receptor. When we knock it out, it blocks endocytosis, a single receptor. When we knock it out, no endocytosis occurs. It has a single transmembrane domain and a bunch of surface beta propeller structures. It looks a lot like a scavenging receptor in our cells. Those things are called scavenging receptors because they have no specificity that people can nail down. They seem to be able to bind a whole bunch of stuff. Our thoughts are that maybe that's what that thing is doing, that it is nonspecific. It is just trying to grab whatever is available and pull it in.
Now, is this cytostome, this feeding structure, present on all stages in the insect host and in the mammalian host, or?
Right. No, it is on all the dividing stages. So when it's replicating, it has it. So the amastogotes inside of host cells have it, and the epimastogotes inside of the insect intestine have it. But the infectious forms break it down and get rid of it completely. So the trypomastogotes don't have one.
Okay, so they're just running on stored energy until they invade a cell?
It appears so. they seem more just like infectious missiles that are kind of destined.
Yeah, they have a very different goal.
A single goal. But
Exactly.
But, the truth is we have been pursuing kind of an understanding of why the cytostome is even there, because we've been able to make mutants that don't even have the structure at all. And so it's a bit of a conundrum for a long time for us of why this thing is even there.
And the mutants survive.
Mutants have no obvious growth defect at all.
That's fascinating because my next question is going to be, obviously this is cool cell biology, but is it a targetable thing for T. cruzi? But if they can survive without it, then maybe it's not essential. So fascinating.
Well, of course. Well, that has been kind of the bane of my existence for a long time.
Thanks.
Sorry to bring
Thanks
it up.
for bringing that up,
Very
Kim.
sensitive topic.
We can go back to talking about immunology.
Every time I lecture on this, I show these mutants that don't endocytose, and it looks more like we're just cataloging essential proteins for the function of this structure. And on some level, we are extremely lucky that this wasn't an essential organelle for a while. So when we first started studying this, T. cruzi, we were just being able to knock out genes. There was no conditional knockdown system available. And so if it had been essential, we wouldn't have been able to study it anyway.
Yeah.
And so in many ways, it was the biggest gift of all that it wasn't essential for us, because what it allowed us to do was to begin to kind of cut our teeth on developing an array of molecular tools to actually dissect this parasite. Because without a conditional knockdown system, you can't study anything essential, and you're stuck looking at things that don't matter. And to me, there's no fun in that. And so by having this system that had a functional output, so we could knock out genes and look at, this thing doesn't endocytose, or it doesn't endocytose as fast. We now had a system where we could say, let's start playing with some conditional expression tools to regulate a gene's expression, and we won't be affecting parasite viability in the process. So essentially, we had non-essential genes that also had a functional readout. So we do these feeding assays where we feed them fluorescent BSA, and we can do a flow assay so we can quickly measure how fast or efficient some mechanism is working, like endocytosis. And so because of the cytostome, we were able to kind of develop a variety of tools, including our hammerhead conditional knockdown system that we published earlier this year. And it was really a result of us being able to fine-tune and fiddle with things for a while with this non-essential organelle. Now, without that reality, I don't know what we would have probably gone on to study something else, if it had been essential. It would have been tricky. We would have just been tagging it and just cataloging things that localized to it. So it was really fortuitous that it wasn't essential. But I think that still brings up the question of, if you can get rid of this structure, is it really something we should study at all if our goal is to try to combat this parasite as a disease, right? And of course, we work for the taxpayer who is looking for us to try to make advances in human health. And it's a difficult sell to be like, this doesn't matter at all. Please fund this. And so I understand the problem. And so that has led us to continue to search for why this thing exists. And so that has been a really cool journey on its own, because for years we tried, looking for nutrients that maybe the parasite needs to get through the cytostome, and nothing really kind of popped out as obvious to us until fairly recently, through kind of a side study, we began to realize that endocytosis can be tuned and influenced by a certain metabolite in the media. And this kind of came really by a kind of a completely harebrained hypothesis that turned out to be wrong, but it really led us to understanding how the cytostome, what it's really for. And so we were doing some studies titrating in a component of our media called heme, which we use a form, it's called hemin. It's an oxidized form of heme. Now, heme is a necessary metabolite for Trypanosoma cruzi. They are auxotrophs, as are all kinetoplastids. None of them can synthesize heme. And this, of course, brings me back to the very strange coincidence that the first lab I ever worked in.
Waiting for it.
It's all coming.
This is where we test if people were paying attention.
It's all coming together.
It's super weird, right? It is very strange that the first thing I ever really studied in the lab was heme biosynthesis. And now I'm back to studying it, which I never really had any thought that I would ever do that again. And we noticed this very strange phenomenon that endocytosis could be tuned by heme. So as we added in more and more heme into the media, endocytosis would go down in its rate. And then as we was like, well, that's strange. Maybe it were some toxicity problem. We're messing with the membrane, even though they grew just fine. And they'll grow in over 1 millimolar heme and as if nothing's different than the normal media, which is like 25 micromolar heme. So we started then titrating the heme levels down. And what we found is as we approached zero heme, the endocytic rate would skyrocket. And I never seen anything kind of increase endocytic rates before. We'd only kind of found mutants that would block endocytosis. And so this struck me as both fascinating and sadly obvious, because we were looking for something that the parasites were potentially auxotrophs for, and this is clearly something they're auxotrophic for. But it never occurred to me that it would be something as simple as heme being the sole reason for endocytosis. So what we've done since is we've identified a heme transporter that's localized to another area of the membrane where metabolites can pass through, and that is the flagellar pocket. And so now things are starting to become clear for us that endocytosis is a conditionally essential or redundant system for the uptake of heme. Now, when we knock out this transporter at the flagellar pocket, now endocytosis becomes essential. And so if we block endocytosis and this heme transporter, the parasites die. It's somewhat painfully obvious that it took us this long to identify this, but people who have been studying heme uptake for a long time in T. cruzi had shown that there were transporters that went to the pocket, and I just assumed, well, that's something independent of endocytosis. Why would you construct such an elaborate structure for something like heme? Yeah, so what we've begun to appreciate is that it has redundant systems in place. Now, the deeper question for me is, why have these two forms of heme uptake? And I think this maybe pertains to their lifestyle. So Trypanosoma cruzi, when it's in the insect intestine, is living essentially in a bath of actively digesting blood, right? Since they live in a blood-sucking insect's intestine. So what we began to realize is, when the bug takes a blood meal, begins digesting red blood cells, releasing hemoglobin, digesting that to release heme, and then there's tons of free heme around in the insect intestinal tract. It maybe can easily import across its plasma membrane through these heme transporters that free heme. However, there are likely times in other parts of its, maybe prior to the digestion of the blood meal, or maybe even when it's inside of a mammalian host cell, that free heme is less readily available. And at that point, because our cells do not let free heme just float around. It's a very dangerous molecule. It creates a lot of reactive oxygen. And so we sequester it inside of proteins, either bound covalently, like in hemoglobin, or loosely, like in a bunch of hemoproteins that act as carriers. And so maybe at times you have to take in actual hemoproteins and digest them to liberate that heme from them. And so maybe one version, say at the flagellar pocket, takes any available free heme, and then the endocytosis maybe is tuned to be able to take in heme that's already complex with some protein and liberate it from it. Maybe that's why these two systems exist.
You mentioned what happens in the insect vector, and that brings up the next part of your lab projects that I wanted to talk about, because you've started, as a molecular parasitologist, you've gotten into rearing one of the insect vectors for this. And how did that project come about?
No, that's a great question. So It began, I guess, with the cytostome problem, is that we couldn't identify a growth phenotype for why we could knock out the cytostome. What is it for? And it got me thinking about other kinetoplastid parasites. The vast majority of trypanosomatids are monogenous trypanosomatids of arthropods. And the vast majority of those are actually fecal orally transmitted from insect to insect. In many ways, Trypanosoma cruzi is kind of an evolutionary oddity in the sense that, unlike its other cousins, the salivarian cousins like Trypanosoma brucei and leishmania, it never learned to go up through the mouth parts and infect via them. The mouth parts are the salivary glands like brucei and leishmania. It still, in many ways, is a fecal oral transmitted parasite, except that it just goes through a long route through a mammalian host, because you still get infected through the feces of the reduvid insect.
Yeah, I've always thought, adding insult to injury.
I agree. It's a terrible parasite. But if you look at all the other monogenous trypanosomatids, all of them have a cytostome, but the salivarian parasites do not. Just logically, it suggests that maybe something about life in the intestinal tract of the insect requires a cytostome. And so I initially started talking with an entomologist here at UGA named Kevin Vogel, and working with him, trying to do some of these experiments. And it was a great collaboration. But I also had my own interest in learning more about how to rear and do the husbandry of the insects myself. And so he was very instrumental in helping us initially set up our colony. I was very fortunate to be able to recruit a postdoc who was a student in Mike Strand's lab, Dr. Ruby Harrison, to kind of help lead that project, since I wasn't an entomologist. But when you start something new like that, you can learn a lot about a different field. And so it was very exciting for me to kind of learn about how the physiology of insects. I didn't know anything about insect physiology. And so when we would start dissecting them and looking at them, it was a whole universe for me of different. When you look inside of the organs of an insect, it is very alien, and it is nothing like anything I've ever seen before.
Are you talking about the Malpighian tubules?
For example.
Yes.
Oh my gosh.
That's the only internal organ I know. Okay. So,
yeah, it's all. Their version of a heart is very weird. Like, they don't really have blood, right? They have hemolymph, so they don't. And then they have these, tubes that go through every single part of their body. Since they don't have lungs, every single cell touches the air through these tubes that go throughout their entire body. And learning about how they grow and how they develop was very fascinating to me. Nothing I'd ever seen before. And then learning about, how to grow them, how to deal with their developmental cycle, how to, feed and house and deal with the husbandry problems was a great problem to get involved in. And that's something that has been a lot of entertainment for me to kind of get into and learn about. And then having an entomologist in the lab has been extremely helpful, because the million questions I have someone that can help me answer them.
And you guys weren't just learning about it. You were developing new methods for growing them, because I would see Ruby with 3 iterations of a 3D printed device to heat the blood meal or for the insects to lay their eggs on, and all these iterations of this. So I know you guys have been perfecting this.
You never underestimate laziness as a vehicle for creativity. So yeah, a lot of these things, I've enjoyed that, and Ruby has been instrumental in helping us design these things. But a lot of these things were the result of my own kind of frustration in doing the husbandry myself. So there were times when it was necessary for me to go in and feed and change and do all these for, there would be certain periods where I was one that was in charge of doing that. And it would just take so much time. And, one of the earliest times I started working on these things, I'm in this thing we call the bug ranch, which is something that was a place where we hold them. It was designed by Kevin Vogel initially. And I would be working in this containment unit, and these bugs, and I'm not a big fan of bugs, to be honest with you. Strangely enough, I do not like them. And so I would be handling them, sorting them in various developmental stages, and they would start crawling up my arm. And I'm like, this is insane. Like, I can't have these things crawling on me. And of course, they're, blood feeding insects. So I'm like, what, are they going to get on me? And I'm going to carry them out and they'll bite somebody. I was like, we can't have this. And so I was like, I can't work with them when they're all creepy crawling all over me. And so I was like, there definitely has to be a better way. And so I started researching what people in the study say flying insects do. And the Drosophila people had already come up with this. And they will gas their flies with CO2, and then they'll go to sleep, almost instantly and be asleep for a while. And so I was like, well, I got to do that. So we bring in a CO2 tank, and that has helped streamline a lot of the sorting and the more cumbersome work. I would spend hours taking apart the cages, cleaning them, and changing the bedding all the time. And I was like, this is taking me all day to do this. And I was
so you're the maid for the insect colony,
right? The bug ranch. Yeah, he's the maid at the bug ranch.
So I was like, there has to be a better way to do this. And so I started looking into streamlining the process and figuring out, instead of using kind of the Nalgene bottles that I would have to just constantly wash, we started using kind of these disposable deli cups, and then these lids that could be clamped down onto these deli cups. And then, Ruby really took charge of developing different iterations of how to make these lids that were, could clamp down and were secure on top of the deli lid. She also started designing, we used to use cardboard for them to crawl on the inside, but she made a 3D printed version of this that had silver embedded ions so that it would keep down bacterial levels and mites as well, which are an issue. And so thankfully, knock on wood, we've had really good success in our colony. It's been extremely healthy. A lot of people's colonies undergo periodic collapse from pathogens that spread through the colonies, which is, common when you rear these creatures in high density. And so we've been, very fortunate to, I think these methods we've been developing over the years have led to what I think is a healthy, vibrant colony that we have very little issues and takes a lot less time and effort to maintain. And so, yeah, like the hand warmer thing, we kind of took hints from other groups that had done quasi-similar things, and we developed these 3D printed holders for hand warmers, and then also using kind of those childlike water balloons that autofill up. We use those to fill up our blood. And now we can feed, an entire colony in one go rather than, when I used to feed them, and Ruby can speak to this way more than I can, but I would spend all day and night feeding, just to have one feed in per week. And I was like, I can't spend 18 hours in here doing this. Like it's way too much time. And so, but now we can do the entire colony in one go, and it's a lot more efficient and not as time consuming, because actually, I'm not in the business of rearing bugs. I actually want to do experiments.
Well, I guess one thing that we should end with is, now that you have all this free time because you've become more efficient, what are you doing in that free time?
Good segue.
On my free time, I just had another child. So I just had a baby boy about a year and a half ago. So he keeps me busy, and I have 2 older kids. So yeah.
That explains where all your time goes.
That takes, believe it or not, children take up a lot of time. Mm-hmm. They are. No, but that's where I'm at when I'm not in the lab.
Okay. Sounds good.
Our guest today has been Dr. Drew Etheridge, Associate Professor in the Department of Cellular Biology and a member of the Center for Tropical and Emerging Global Diseases. Drew, thanks for being with us.
Thank you. It's been a pleasure.
Links to additional information about Dr. Etheridge's research can be found on our Instagram account, which is at PPPPodcastUGA.
As always, we appreciate you tuning in today. And if this is your first time listening to the People, Parasites, and Plagues podcast, we encourage you to check out the other exciting episodes launched last season and this season. Please rate the podcast, and if you have any questions or suggestions for future podcast episodes, reach out to us via email at ppp@uga.edu. This podcast is brought to you by the Faculty of Infectious Diseases and the Grady College of Journalism at the University of Georgia. It is supported by the University of Georgia through the Office of Research, the UGA Graduate School, and the College of Veterinary Medicine. Special thanks to our production assistant, Justin Nemitz, for research, editing, scheduling, and keeping us on track.